Hello, I'm Clyde Wilson. Thank you for this invitation to speak on the role of the super-relaxed state of myosin in human metabolism. This is work done by myself, Narman Naber, and Roger Cook in the Department of Biochemistry, Biophysics, the University of California, San Francisco. The super-relaxed state of skeletal muscle lowers resting metabolism by as much as 1,000 calories per day, contributing to the very large dynamic range of resting metabolic rate, and therefore provides us a pathway for increasing metabolism, thereby reducing metabolic disease risk significantly. So what is it? How is it measured? And how can we regulate it? In a nutshell, the super relaxed state is destabilized, increasing metabolic rate through three known pathways so far: phosphorylation, piperine, and calcium. Phosphorylation occurs on myosin with twitch potentiation, which keeps calories burning at a higher rate for many minutes after force production in muscle. Piperine, which is the active compound in black pepper, destabilizes the lower metabolic rate of myosin, thereby doing the same thing to the same extent, as well as calcium, which destabilizes the super relaxed state, increasing the metabolic rate in skeletal muscle as well. So myosin is a very large protein with two heads that are bound together through an alpha helical coiled coil. And these two heads can be bound to each other and back against their own thick filament backbone that holds them in a helical array, so that they're effectively in total shutdown, burning very few calories, less than 100 per day. You can think of this as a car whose engine is completely shut off. That's the super relaxed state. You'll increase the calories to 1,000 per day if you keep that engine idling or running, even though it's still at rest. This is the disordered relaxed state, where the heads are free from their thick filament backbone and idling and burning calories, but not bound to actin. With a nerve signal releasing calcium from the sarcoplasmic reticulum and skeletal muscle, the calcium will, through excitation contraction coupling, uncover the binding site on actin, so that these idling heads can finally bind and generate force. So here's a structure of the thick filament, whose backbone is made by the tails of the myosin molecules themselves. And you can see this helical array around that thick filament backbone of pairs of myosin heads binding to each other and back up against their own thick filament backbone, parked in full shutdown. Two of those myosin heads with their necks coming off of those heads are shown here in yellow. And there, here to the left, is a space-filling model of those two myosin heads, blue and green, each of whose neck linkers have two smaller proteins on them, the light chains, the essential light chain and the regulatory light chain. For the green head, that's in pink and salmon colors, and for the blue head, yellow and white. In species like tarantula, where they lay in wait for days at a time waiting for food and could starve to death if they didn't have a way of shutting down muscle to the extreme extent, the super relaxed state of burning fewer calories is in this interactive heads motif, as it's referred to, or IHM. This interactive heads motif, where the heads are interacting so strongly with each other in their own thick filament backbone, turns over ATP in the order of every half hour or more. But one of these heads, the one colored in blue here, is more blocked than the other, because the green, the free head, is partly bound over it, locking the blue one in place. So the myosin-free head will swing out and back into the interactive heads motif. And in the time that it's swinging out, however brief that is, many more ATPs are being burned by that head. So instead of the ATPase, or the lifetime for ATP turnover in myosin being on the order of every half hour or more, that ramps up all the way to a few minutes, roughly 230 seconds, depending on whether you're looking at mouse, rabbit, or human. If you destabilize the superrelaxed state, it keeps that free head and ultimately the blocked head out of the interactive heads motif, so that you're now in the disordered relaxed state. So this is where you're disrupting the superrelaxed state with phosphorylation, calcium, or piperine. And the ATPase, or the rate of calorie burn, goes up by an order of magnitude. That's where your resting metabolic rate is now 1,000 calories higher than if you were in the superrelaxed state, where the free head is just briefly swinging back and forth and burning 10 times fewer calories. So this regulation of the superrelaxed state, or destabilizing it to increase resting metabolic rate, occurs here in the regulatory light chain. So here's an expanded view of a model of the regulatory light chain structure on myosin, which is the white coil here coming through the regulatory light chain. And I placed phosphorylation, piperine, and calcium very approximately in their interaction points, where they're converging on this regulatory light chain to change its conformation to destabilize myosin. So how do we measure it? ATPase, or the rate of ATP turnover, is generally measured by the buildup of turnover products. But because the disordered relaxed state turns over ATP so much faster, that ends up being the elephant, where you're trying to measure the weight of the fly, the superrelaxed state, much slower on top. So in order to overcome that barrier of having it washed out by the high ATPase of the disordered relaxed state, Roger, almost 2 decades ago, in other work, developed a fluorescent analog of ATP, MANT ATP, which you can then have bound to the nucleotide binding pocket of myosin, which then gives the fiber its fluorescence. You can see in this left screen here the fluorescent fiber with the fluorescent myosin in place, and wash that out with ATP, reducing the fluorescence. You can see the decay curve here. That gives you a time resolution so that after the first 20 seconds, you're able to resolve the super relaxed state. So this is how that time resolution was achieved, to finally be able to measure the super relaxed state. So in the experiment, we put 2 regions of interest where we're measuring the fluorescence on the fiber, and 2 background regions of interest. You can see the higher curve here on the right is the decay of the fluorescence as the fluorescent ATP is being chased out by ATP, and then beneath that, the background. And our fiber is sitting here on this slide in this simple flow cell on the microscope stage. So now I'm going to expand these screens on the left and on the right. Here's our single skinned fiber, skinned so that the ATP chasing out the fluorescent analog is able to freely diffuse into the fiber. Here's our regions of interest where we're measuring our fluorescence compared to our background. And then what you get is your fluorescent decay with two main terms, because in mouse, rabbit, and in human, we don't see the half-hour plus lifetime version of the super relaxed state that we refer to as the long super relaxed state. Instead, we only see the short super relaxed state, where the free head is going in and out of the interactive heads motif. So here's at the front end of the curve, most of our fluorescence loss is from the disordered relaxed state, where ATP is being exchanged out very quickly. And then after that is completed, after roughly the first 20 to 30 seconds, that's where the curve, the shape of the curve is then dominated by this longer ATP turnover time of the super relaxed state. This is the publication first publication and demonstration of the existence of the super-relaxed state, which was in rabbits, done by Melanie Stewart in our lab, published in the Proceedings of the National Academy of Sciences in 2010. Leonardo Nogara, then subsequently within the lab, tried to see if we, using over 2,000 different FDA-approved compounds for human consumption, if any of them destabilize the super-relaxed state, increasing metabolic rate, and in microwell arrays, had one hit out of over 2,000 compounds, which was piperine, the active compound in black pepper. The lab got the cover of Proceedings of the National Academy of Sciences with that paper, and it had been known, piperine, to reduce the extent of obesity and type 2 diabetes in rodents for decades, but until this paper, there had been no hypothesis on the underlying mechanism for that effect. So as part of Leonardo's work on this project, he showed that the KD is about 3 micromolar for piperine. So you can see, at 3 micromolar, you're halfway up to the plateau, and then at about 20 micromolar, you're plateaued at your maximum ATPase, and that matches identically to the destabilization of the super-relaxed state. So the higher ATPase, or calorie burning rate, incurred by piperine is due to the destabilization of the super-relaxed state into the disordered relaxed state. So where you're parked to where your car is idling. But with such a low KD, or binding affinity, it would take 1/4 of a cup of ground pepper per day just to initiate a response. So what this demands is analogs with a higher binding affinity or specificity to the regulatory light chain of myosin in order to be an effective pharmacological agent. So then Leonardo showed the same fluorescence decay curve with the faster and the shorter ATPases within myosin, demonstrating the super-relaxed state and its effect of piperine. And that curve looks exactly the same as the result in human that myself and Narman and Roger just published a few months ago. And so I'm just showing you the human results here, where without piperine, you can see that the super-relaxed state persists longer. That's the higher line, the higher fluorescence. So ATPA, the fluorescent analog of ATP, is being exchanged out slower, as opposed to with 100 micromolar piperine, where the MANT ATP, the fluorescent analog, is exchanged out faster, and there's no statistically significant effect on human cardiac muscle, which is an important for drug development, because that would be a unacceptable side effect. So in comparing lean to obese humans, 3 subjects of each from the tissue bank at the University of Kentucky, which vastus lateralis biopsies, the super-relaxed state population and lifetime were not statistically different. And for both populations, the reduction in the super-relaxed state with the application of piperine was about 25%. So in the right direction of drug discovery, giving us a pathway, but needing a more specific binding. With muscle activation, there's calcium release, which binds to calmodulin, activating myosin light chain kinase, phosphorylating the regulatory light chain of myosin, the same part of myosin that piperine acts on. And Melanie had shown in the previous work that we discussed just a few slides ago, that with increasing regulatory light chain phosphorylation on myosin, you have a successive reduction in the super-relaxed state population. So Nariman in the lab has, with the more traditional measurements of ATPase turnover, shown that when you stabilize the super-relaxed state, or the interactive heads motif with blebistatin, that you suppress myosin ATPase, and that phosphorylation increases the ATP turnover, or rate of calories, metabolism in muscle to the same extent as piperine, and that these effects are not additive between the two. It had been shown decades earlier by Houston and coworkers that with a 10-second maximum voluntary contraction, you phosphorylate both fast and slow twitch fibers by a factor of 2, and that phosphorylation is sustained for 10 to 30 minutes afterwards. So this is pulling myosin out of the interactive heads motif, and where the free head is swinging back and forth with a low calorie burn, to where the heads keep idling and are waiting for the binding site on actin to be uncovered to generate force. So this is our twitch potentiation, and you're increasing your resting metabolic rate in that state by an order of magnitude. So a hypothesis would then be, if you were to move throughout your day every 10 to 30 minutes, that you could keep your muscles idling and dramatically increase your resting metabolic rate by moving at that periodicity. So in fact, we do see that what's referred to as the excess post-exercise oxygen consumption, or the increased calorie burn after movement is completed, is significant. So with a half hour of walking, that's the red block, this is looking at calories or energy utilization in humans. This thick black line on the top of the red block, after 30 minutes of walking, you're burning about 20 calories. But if you then walk for 2 minutes every half hour, so that the total amount of walking through your day is the same half hour, but it's broken up throughout the day, each of those 2-minute walks has the same calorie burn afterwards as if you'd walked for a half hour. So you keep burning 20, so you use up 20 calories walking, you burn 20 calories after you've stopped walking. And you keep repeating that, and you end up with as much EPOC, excess post-exercise oxygen consumption, that's this black box here, as the calories used up in walking itself. And if you do both, a half-hour walk and broken up 2-minute walks, all of the extra calorie burn comes from those 2-minute walks. Because it's the same after 2 minutes as it is after 30. In human diabetics, if you move or walk for a minute or two every 1/4 of an hour, as opposed to every half hour, as opposed to every hour, you get successive improvements in stabilizing your blood sugar, lowering your blood sugar after breakfast, lunch, and dinner, with a reduction in overall insulin throughout the day, and a reduction in total glucose area under the curve over the course of roughly your 24 hours. And in human diabetics, if you do a half dozen 3-minute walks through your day, even if those walks are compressed into a 6-hour time period, these diabetics are seeing hyperglycemia being lowered into normal glycemia, not just during the day in the time period that they're doing these activities, but all the way through 24 hours through the time that they're sleeping to the next morning. So this helps us to explain the powerful benefits of non-exercise activity thermogenesis, or NEAT, against weight gain from overfeeding by as much as 1,000 calories a day, where the more non-exercise activity thermogenesis humans will experience, the less weight gain, all the way down to 0, from overfeeding. On the flip side of this, metabolic adaptation, even 6 years after weight loss, this was shown with Biggest Loser contestants, 6 years after being on that competition, can approach 1,000 calorie a day theoretical limit of the super relaxed state. So here you see in the 30-week time period of being on that television show on The Biggest Loser, with very intensive exercise and strict dieting, that on average, they have a metabolic suppression of about 500 calories per day, some of them as much as 800 calories a day, which is approaching that theoretical limit of the super relaxed state. So could this be epigenetic changes, post-translational modifications? We had 3 subjects in lean versus obese, and we're not able to resolve a difference. You could see a trend towards that, but we would need a much larger population to move in the direction of being able to resolve that, and how it's different between individuals. So the super relaxed state helps explain the decrease in metabolism seen with weight loss, and an increase in metabolism after brief movement. And comparing regular brief movement to continuous exercise, the calories burned after movement can easily be doubled relative to the calories burned during the movement itself. And meta-analysis published in 2020 shows that for energy matched regular movement through the day versus one continuous bout of exercise for the same number of calories, that there is a significant improvement in lowering by 10%, about 1/2 standard deviation, fasting triglyceride, glucose, and insulin levels. So what's next? We're looking at the fact that calcium raises ATPase by binding to the regulatory light chain to the same extent as piperine and phosphorylation, and is therefore an additional pathway for getting a handle on the regulatory light chain for increasing metabolic rate. You can see that the effects of calcium and piperine are cooperative. And so in summary, the super relaxed state of myosin helps explain the wide dynamic range of metabolic rate, including the large potential loss in resting metabolic rate during dieting and or exercise, the large benefit of regular non-exercise movement throughout the day, which I highlighted here in purple, as well as the phosphorylation and very roughly where it interacts, and gives us targets for pharmacological pathways through piperine and also potentially through the calcium binding region, which I color-coded in gray. So myosin is, in a sense, myosin's regulatory light chain, using the car metaphor, idling is our potentially our metabolic steering wheel. Thank you.